this computer. Perfect. Okay. So now let's get into viral pathogenesis. Okay, so good morning, everyone. First of all, I hope everyone's doing safe. Now we're going to go back, let's say, into virology. The next day, I think we're kind of, let's say, done with immunology. I think that's kind of the most of the immunology that we're going to be giving this year. Now we're going to be more focusing on more about viruses, but also it's going to be a nice, let's say, it's going to be nice to see the combination of virology and immunology in the next couple of chapters. And to really, let's say, where would you see viruses and immunology coming in together? It's especially when viruses start to attack us and try to kill us. And that's what viral pathogenesis actually means. It literally means viruses causing disease towards us. So what is viral pathogenesis? Well, viral pathogenesis, it really refers to the whole process in which a virus comes in, it infects us, and it causes a disease. Hence patho coming from more disease. So today we're really going to be talking a lot about all this entire process and how it starts. But as we know, it always starts with the virus entering us, and it has to go to a host, because we know viruses are obligatory parasites. They cannot live on their own. They need to be infecting some sort of cell in order to keep replicating, because they do not possess the machinery to replicate and do multiple copies. So they need to go inside a living cell that actually has machinery to copy its genome, to produce its proteins and its glucans and glycans, all this different stuff. So when it comes to actually entering it, we can see basically two things that can happen at two levels. Well, at the first level, we can see that the virus is infecting a cell. It's hijacking its machinery and it's altering its old functions, and this can lead to death, to the death of the whole cell. At the level 2, of course, you're looking at this at a cellular level. But if we zoom out, a lot can happen basically on a human level, on a whole, let's say, or as a patient level. And of course, this could technically be meaning the immune response that we're getting, the spread of the virus, the symptoms that we get. So one level is what's happening at the cell. What is happening inside the cell host? Like, is it snatching some certain proteins? How is it affecting the whole cell's function? And the second level, which is looking at zooming out a bit, what's happening to the patient? Is he getting symptoms? Is the patient dying? All the different things. And these two levels, despite them being in two different, let's say, kind of levels in some sort of way, they're very interconnected with each other, and it makes sense, because whatever happens to the cells is going to happen towards the patient, because cells make up tissues make up organs. So if a virus is able to kill quite an abundant amount of cells, it can really damage the tissues, which can then damage a vital organ like the heart or the lungs, which can really have a huge impact. So the whole, let's say, outcome of a patient really comes between how a virus is interacting with a cell. And today we're really going to learn a bit more about that. How does a virus interact with a cell, and how can that lead to certain different outcomes towards level 2? As we know, let's say the central battlefield of basically when it comes to a virus versus a human, it always starts in a cell, because the virus, as we said, is a kind of obligatory parasite. It has to go inside a cell to start live, to replicate itself. Once inside the cell, four outcomes can come up. One is cell death. Second is fusion with the neighbors. There should be 3 over here, malignant transformation, and 4 should be survival while producing viruses. So either the virus comes in and it literally kills the whole cell, or it actually makes the cell fuse with other neighboring cells. We've spoken about this. This is called synthesia, which technically is forming a massive giant cell that is literally the formation of multiple cells fusing together. Malignant transformation, it actually makes the cell cancerous. For some reason, viruses have a very good, let's say, ability to go inside the whole cell, the host cell, and turn on certain machinery to make it divide uncontrollably. And that is basically cancer. Or the virus can actually stay there, not doing anything towards the cell, and just keep producing. You know what I mean? They live in this, let's say, symbiotic relationship. The virus is there, is just producing, and is letting the cell do its thing. So these are 4 different outcomes that can happen. What outcome comes up? It's not random. It's specific, very specific on how the virus interacts with the host pathways. How does the virus actually manage to change the cell? Because as I said, viruses, for example, can turn on apoptotic machinery inside the cell. It can induce the cell to die because for it, sometimes the virus wants the cell to actually pop because the dying of the cell will release all its virions so it can infect even further. For some other viruses, they don't want that. They want to stay inside the cell and they want to basically make it control uncontrollably. So it's going to make it cancerous. Papilloma viruses is, for example, one of those. In the case of, for example, herpes viruses, they really do not want to kind of do a lot of killing. They sometimes they want to stay kind of hidden from the immune system. So what they do is they keep inside the cell and they don't do anything. So the outcomes depends on how the virus interacts with the cell. It's not these outcomes are not random. It's all based how a virus really interacts. And of course, understanding how a virus interacts then with the cell tells us about a lot about the disease and the tissue damage. Because will you get cancer? Depends on how the virus reacts towards the cell. Will you actually, let's say, get blisters, for example, from herpes? Well, depends on how the herpes interacts now with the cell. Will it start destroying it? Will it start causing all this inflammation? Well, yes, then it's gonna cause the blisters then. So now let's talk about all these different things, like these different outcomes. So now it comes into viral-induced cell death. As we know, a lot of viruses can actually really induce death towards the cell. It's quite well known. But how does it do it? Well, many times it can actually be the thing of viruses in some sort of way inducing apoptotic pathways. But most of the cases, it's usually viruses actually inducing or stopping the host cell's ability to produce proteins. If you look at about a cell, sometimes when you look at a certain thing, what makes a biologist define that a cell is living or not? What's then the characteristics of that? It's simple. Is it producing proteins? If a cell is producing proteins, we determine it as living. If it's not able to produce proteins, then no. That we say is technically dead. So what happens technically with a virus is a virus is going to come inside the cell and it's going to stop it. It's going to literally rob its machinery, specifically ribosomes. It's going to rob the ribosomes or rob other machineries that are literally all needed for translation or transcription. So that is what makes cells sometimes die because of viruses. Because viruses come in and they literally interfere with the production of macromolecules, specifically proteins. Because as a cell to be living, it needs to produce hormones, it needs to produce a lot of proteins for cell structure, a lot of different regulator signaling proteins to keep things in order and check. Well, now the virus is robbing that. It's stopping the host cell to do all these very critical functions, and this can lead to death. And that's usually what happens technically. Give you an example of how viruses are able to do that. Polioviruses. Polioviruses love to do this. What polioviruses do is they come in and they actually sequestrate or inactivate this host initiation factor. So initiation factor, AIF, is a factor. Most probably you guys have, learned this before in some other class. It's a certain factor that comes in and it starts literally the whole translation. It binds to your mRNA along with your ribosomes and everything, and it literally starts this whole process of translation. It's needed. Without your initiation factor, translation cannot occur. So what happens here is that poliovirus comes in and it steals it. And it doesn't allow it to come basically bind to your mRNA and your ribosome. So it actually stops the host's way of translation. But what poliovirus does also very well, polioviruses has something inside its genome called IRIS. It's called an internal ribosome entry site. And basically it's found in your mRNA. So this IRIS, what it does, it binds to ribosomes without this initiation factor. So poliovirus from one side is inhibiting the initiation factor from doing anything. It's just making it inactive. From the other side, it's like, okay, fine. We don't need basically the initiation factor because we have our own initiation factor, which is IRIS. So it's stopping the host's way of translation and doing its own then translation. And that's why poliovirus is extremely successful at killing cells because it's able to stop all of translation. Poliovirus is one of the most successful viruses actually in replication because of this IRIS. And because this IRIS is so important and so, let's say, effective at inducing translation in molecular cloning, every time I want to clone, let's say, a protein, I use IRIS. Every time I have my protein, I put an IRIS site because I know if I put an IRIS and like an IRIS site, it's gonna bring all the ribosomes and suddenly my translation is gonna be increased at least 10 to almost 50-fold. So this is how good this IRIS is that to this day, we're still using it in cloning proteins if you want to have a massive production of proteins. But one thing that we see also during viruses basically replicating quite a lot is this inclusion bodies. And inclusion bodies are literally discrete intercellular structures where you see a lot of viral proteins and virions. So when a virus comes into the host cell, it's going to start replicating like crazy. And what you sometimes see with certain viruses are these inclusion bodies. These are just examples of what you see. This is called a negative body. It's an inclusion body that's basically from rabies. So rabies, when they come inside, they replicate so much, they create these big inclusion bodies called negative bodies. And there's literally, it's viral proteins or viruses literally in there being formed. This is with CMV. We call it owl's eye because it literally looks like an owl's eye. And it's very beautiful to see basically in a microscope. But these negative bodies, or sorry, these inclusion bodies can occur, of course, in the cytoplasm or in the nucleus. All depends where the virus replicates. So these inclusion bodies can be very useful for doctors because this can help a lot in diagnostics. Because sometimes we know that someone is actually, let's say, infected with a virus, but we don't know what virus it is. So by sometimes looking at the cells, like if you take a bit of a biopsy and you look at the cells, if you look at the cells and you see what's happening in there, you can sometimes see basically what virus is replicating by looking at the negative bodies, about the inclusion bodies, because every inclusion body for every virus is different. So you see that negative body here is very different to the owl's eye. So some viruses have a very specific inclusion body. They have a specific, let's say, size, shape, location. And by looking at the inclusion bodies, you can actually determine what virus are you being infected with. So diagnostically, it can be extremely useful to know. One thing, as we said, that can happen when viruses start to replicate other than death is syncytia, cell fusion. This happens a lot with herpes viruses, but also paramyxoviruses like RSV, respiratory syncytial virus. RSV, by the way, is one of the most common viruses. By the age of 2, everyone has been infected by RSV. It's a very common virus that infects us when we're like usually very young, very harmless. If your immune system is quite powerful, is competent. If you have issues with your immune system, it can be very deadly. So in these cases, the herpes viruses, the RSV, what they do is once inside the host cell, they will start to express their own receptors, so viral receptors on the membrane of the host. So these yellow things over here, so a bit small, but these ones with the arrows, these are viral proteins. And what these viral proteins will do is they will literally bind towards the host cell, other host cell receptors, and induce this fusion. You will get this fusion, and you will create this massive multi-nucleated cell. We call these giant cells. Why would a virus want to do that? Well, viruses like herpes and RSV, they don't want to be seen by your immune system. They want to hide from it. So the best way of hiding from it is to actually stay inside. Because, for example, antibodies are not able to go inside a cell. It's only able to bind to outer membrane, or basically proteins that are outside, not any intracellular proteins. That they don't do that. So a virus being inside, it kind of protects them from antibodies. So of course, a virus inside one cell, it comes to a limit how much they can replicate, how much machinery they can take. But now with the fusion of all, let's say I have now 5 literally cells, you have so much more machinery, so much more space for replication. So the virus doesn't need to really escape or go outside. See the chat, there's a question. But synthetium is an abnormal structure. Isn't it detected by the immune system? That's a very good question, Miriam. Yes, to a degree, yes, it is actually seen by it, but not to it to a degree. It is seen because, of course, there still is a lot of things. There's during this formation of synthetia, there is a release of DAMPs. Some intracellular proteins are released to the outside, and those are danger signals. But it's not as actually bad as rupturing a whole cell. Because if you rupture a whole cell, so much release of DNA and ATP and ribosomes, all these things that shouldn't be at the outside, that causes a bigger immune response. This formation of this big nucleated cell does induce an immune response, but it's still very little compared to killing it. So basically, you're still inducing it, but to a lesser degree. And this is just an example of, for example, how you see these giant cells. If you look at here, so these are massive cells, like we have multiple nucleuses together. This could be very, again, clinically significant because, for example, what doctors can do is scrape a bit of skin or certain areas where you have an infection. And if you see these big, let's say, cells, you start to see like, okay, these are syncytia, these are giant cells. And this could be, of course, telling that this could be herpes, for example, or this could be RSV. Usually, if you're quite, let's say, older, it's mostly herpes. RSV, you do get it, but it's mostly when you're a child. So these giant cells have a huge clinical significance, helps us a lot with diagnostics. And of course, one thing that we know as well with that cells can do is kill. And when you kill, you have this cytopathic effect. We spoke about this previously. I think it's in like chapter 2 or something. So this is what happens technically with cytopathic effect. So for example, these are A549 cells. These are lung cancer cells that you have in the hood, or sorry, in your lab. What happens if you infect a virus? Like little by little, the virus is going to start replicating, and they will induce apoptosis because some viruses will induce apoptosis. So what you have is this, let's say, change, this morphological change within a cell. And for example, if you see here, these are cells that have these very beautiful, let's say, rhombus, triangular kind of look. But bit by bit, their morphology starts to really change. And it's super interesting because I do this quite often, that I infect a lot of cells with my viruses. And you see this plaques, literally this kind of circular kind of regions. It's not that you see one cell here, one cell here, one cell here, and the other one there, that they're dying. No, it's usually a border. They create these beautiful borders. Don't ask me why. I really don't know why you get this, but this is called the cytopathic effect, the CPE effect, where you see these borders over here of this kind of death or this change of basically cells. They're not dead yet, but they're going to die. They're literally, the morphology is changing bit by bit. And this, for example, this could be, let's say, day 0. This usually happens at the first week. This then happens like week and a half or around 2, where now all your cells are literally went from a triangular kind of shape towards a much more circular shape. When a cell is very circular and it's literally not attaching that well, that morphology is already telling you that these cells are not in good shape, they're dying, or they're actually dead. So this is called the CPE effect. And this could be very diagnostic also, the cytopathic effect, because what I can technically do is I can take a bit of a fluid, for example, from a patient, bit of a sample fluid, add it on top of cells. And if I see the cells are having the CPE effect that is very distinctive of virus, I can actually see like, hey, this guy has a viral infection. And of course, you can start ruling out certain things because we know like certain viruses will not do CPE effect, so you can rule them out. So diagnostically, this is extremely useful for a lot of clinicians. But of course, other than just diagnostic, I use this a lot in the lab. I don't know if anyone of you guys have done a plaque assay. Sometimes when I create viruses, I need to know, okay, I create a batch of it. I need to know in my batch of viruses, how many viruses in my batch is infectious. How can I actually know this? Well, what I do is I take, again, a layer of cells, just like this over here, and I add basically different concentrations of virus. So I put, let's say, 20 microliters, 50, and 100 microliters of my batch. And what happens is basically I let it for a couple of days, usually around 2 days or so, because around 2 days, I get this effect. I don't get all my cells dying, I get these basically plaques. And that can tell me a lot about the quantification, how many viruses have. So this is just an example of basically, this is 5 microliters, this is basically 50, and this is basically 100. So you see that at 5, you have very little plaques, at 10, you have much more, and at 100, you have quite a lot of plaques. Of course, to these cells, I add a, let's say, purple dye, or like a purple stain, so my plaques are much more visible. So now the plaques look white in that kind of sense. And by counting the plaques, it can quantify, it is actually proportional towards the amount of viruses that are infectious. So I can really start to see how much infectious viruses I have as a concentration. And that's what we usually do. We usually, for example, in the clinics, when I am injecting viruses towards a cancer patient, so of course, these viruses are very specialized against cancer, by the way. I say inject basically 1 times 10 to the power of 11 infectious viruses per kilogram or something like that. So I usually use plaque assays to determine how much infectious viruses I have. Another thing that viruses are extremely notorious at doing is malignant transformation, so causing cancer. So once what can happen is viruses can go inside the cell, and it's in its own personal interest that the cell starts to replicate uncontrollably. Because if it's doing that, that technically means that the virus is now using all the machinery. Because when you have a cell that's growing, let's say when you have a cancer cells have a huge amount of ribosomes, have a huge amount of amino acids. Their whole metabolism is switched to accommodate for growth. For the virus, that is perfect. That is literally music to its ears because it needs all of these basically ribosomes, amino acids, and metabolites to grow. So sometimes what it will do is actually on purpose make the cell start replicating like crazy. And of course, that can induce cancer. A very good example for this is papillomaviruses, the HPV virus. Human papillomaviruses will start to make literally our cells grow uncontrollably and can cause a lot of cancers. It's the major cause, well, they say it's the only cause of cervical cancer. So 99.9% of cervical cancers come from HPV. Penile cancer comes also from there, anal cancer, but also oral cancer. And you can see here, these are examples of what you see over here. This is basically a patient with oral cancer because it contracted the HPV virus. It's quite dangerous, this virus. And then you have the silent infection. These are basically viruses that they replicate and they do not do any morphological changes. Nothing happens. The virus is there and it's just replicating. And the cell almost doesn't even realize that something is happening, to the point that our own immune system doesn't even realize this. And the virus keeps doing it. And this is just to show that the relationship between a virus and a host doesn't need to be destructive. It can be benign. And we know this for a fact, that there is millions of viruses within us replicating without causing us any damage. There is a symbiotic relationship with more microorganisms. People believe or think that bacteria or viruses are harmful to us. That is not true. I would say the minority of viruses and bacteria are harmful. There's a huge amount of them that are not. These are undetectable by class assays. Yes, correct, Miriam. You cannot, because they don't do any CPE effect. The plaque assay is only for plaque. The plaque assay is only for viruses that do the CPE effect. So these are mostly like adenoviruses. Herpes viruses can also do the CPE effect to a certain degree. CMV viruses, polio viruses, these ones can do this kind of stuff. Silent infections are only usually detected using reverse transcription or basically quantitative PCRs, depending, of course, what the genome of the virus is. But usually, in general, this is just to show that viruses really do live within us. People say that around 2% of our genome is consisting of viruses. A lot of viruses, what they love to do is they come in and they dump all their DNA into our genome. They literally insert their genome into ours. And when we were basically sequencing our entire genome, we started to discover new viruses, actually, by the way, by looking at our own genome, because we started to see like, okay, what is this piece of DNA? And for other further investigation, we started to realize, but this piece of DNA is coming from viruses. How is that possible? And yeah, that's how we started to learn a lot about viruses, by looking at our own DNA, because our DNA is filled with viral DNA. So now let's go into more pathogenesis. So now we know how, or let's say, a virus can do to a cell. Okay, now let's look at it at a bigger level. Let's look at it at a patient level. So as I said, pathogenesis is a multi-step. It's an ordered cascade. It always starts with basically entering a patient, and then infecting a host. It has to go inside the host. And what happens to the host cell, we just spoke about. It could be cancer, it could be death, it could be nothing, or it could be just the syncytia. This is what can happen. And then it starts to replicate, and then here comes the spread to the other tissues. Will it spread to other tissues? Will it just stay within one, let's say, place, or will it just basically be completely resolved? That's where things can differ between virus. And it's not, of course, as I said, the host. So this whole determination how pathogenesis occurs, how severe it is, or how it happens, it can be through viral receptors with host receptors, like how the interaction occurs. But our immune system also has a huge and undeniable, let's say, effect there. Our immune system really can activate or control infection. And the disease outcome, it really then depends on how the virus basically interacts with the host cells, but also with the immune system. So as we said, when it comes to the stages of viral infection, we first have something called the incubation period. This is basically the time between the infection and the onset of symptoms. So if I'm infected, let's imagine with influenza, I'm not going to directly get in symptoms. We call this the incubation time or incubation period, and where the day that I get infected till the day I get a symptom could be 3 days, it could be 4 days, it could be 1 week, or it could be a month. It depends on the virus. It could be even a year that you get the first disease, or sorry, the first symptom. So this usually happens when your virus reaches a certain, let's say, an amount that is able to cause significant damage. Either it's causing damage to the cell or it's activating your immune response, one of them. And once it really hits that first, let's say, symptom, we enter this prodromal period. The prodromal period basically means that very non-specific symptoms. When the virus starts to hit a certain, let's say, threshold in amount, our immune system is going to, of course, detect it. Either the host receptors, so we know that cells have their own certain, let's say, system, but the innate immune system can also detect viruses. Once they do that, they start to go crazy and they start to release a lot of cytokines. And cytokines, what they love to do is they love to induce an inflammation. And inflammation, you get a lot of fatigue and a lot of fever. So this is why we have non-specific symptoms. When a patient comes up to us and says like, hey, I have fever and fatigue, this is the most common symptoms. This could be bacteria, this could be viruses, this could be almost every single type of virus. It's non-specific, and this is purely because your immune system is just causing an inflammation. Then after the symptoms start to fade down, you start to get your specific, let's say, symptoms. These are characteristic symptoms that appear from damage to target tissue. So this is very specific towards the virus. If you have a virus that it's in your liver, then you're going to get basically liver symptoms. If you have viruses in your skin, you're going to get rashes or whatever specific to your skin. So this all then comes to what type of virus you're infected with. And then finally comes your recovery, and where your viral replication starts to decline. This is where your immune system starts to kind of remove your viruses. This doesn't always happen, the recovery. In majority of the cases, it happens, but in some cases, recovery doesn't occur. And we'll be talking about that a bit in a bit. So transmission, as we know, it's the first, let's say, the first thing that has to happen. Transmission, technically. Two ways that viruses can be transmitted: horizontally or vertically when it comes to human contact. Horizontal basically means from person to person. This could be through aerosols, so respiratory. This could be an ingestion, so eating, let's say, certain foods that are, let's say, infected with viruses. This could be through sexual contact, or this could be through bodily fluids like blood. This is what we mean by horizontal transmission, that is going from one host to the other. And this is what we call horizontal. It's going from one person to the other. But we also have vertical transmission. That basically means from mother to an offspring. So this is usually through the placenta, through birth, or through breastfeeding. And we call this basically vertical transmission. This is how human to human transmission occurs. But of course, we're not just limited to human. We also have other animals, other animal reservoirs, we call them, that are basically affecting us. So reservoirs are basically, I'll give you an example of an animal reservoir. Is, for example, you have bats. Bats are reservoirs to, for example, the SARS viruses. Camels were a reservoir for the MERS virus. These are the basically COVID viruses, the corona viruses. These are animals that are harboring these viruses. And the issue with these animal reservoirs usually is that these animals don't have symptoms. They actually just have a massive load of viruses in them with no symptoms. And they're literally in contact with us. And these viruses, they jump from the animal reservoir towards basically the human. And that can be extremely dangerous. This really can be very dangerous because viruses coming from an animal are so different and really can cause, to the point that when it infects a human, two things can happen. A, our immune system doesn't detect it because it's too foreign and it could kill us. Or that our immune system detects it so well because it's so foreign that our immune system goes crazy and it starts to kill us. So usually zoonotic transmissions are quite dangerous. And of course, we also have vectors like mosquitoes. Some basically viruses, they love to live within a mosquito. And mosquitoes, what they love to do is they love to basically literally suck our blood. So every time they're literally pinching us to take blood from us, they will basically leave viruses. A good example for this is dengue. Dengue viruses are a pure example. And of course, now that it's infected, the infection can be localized or systemic. Localized basically means it remains in a certain tissue. It stays there, it's confined, and it doesn't spread. These basically, an example for this is your common colds. This could be your RSV viruses, for example, your rhinoviruses, these ones that stay just there. And these usually are very easy, let's say, to treat because in generally they have a shorter incubation times. Oh, by the way, yeah, they have a short incubation time because usually they're just doing damage to one tissue, so they don't need to spread to other tissues to really do the damage to really then get to bigger numbers. So it's just in one place. So their incubation time is quite slow. Usually it's around 3 to 4 days that you get a symptom. And to actually remove it is quite easy because it's just one part. This is just an example here of this is gram-negative bacteria, but forget about that. But in case of localized infection, you actually have a lot of your immune system that is literally directed towards that area, and you can get a heavy concentration of them there, which really is going to help with literally the removal of your infection, be it a virus or be it a bacteria. So in generally, localized infections are quite easy to treat. The issue comes with systemic, because systemic, this is where the virus starts to disseminate into other organs. So here's an example. You have an infection in your liver that suddenly starts to disseminate to all parts of your body. This usually happens through the blood. So what happens is the virus literally enters your bloodstream and it starts to travel everywhere. Once the virus hits the bloodstream, we call this viremia. Viremia basically means that the virus is in your blood and it's traveling. This is very immunogenic. It causes your immune system to go crazy and can be very deadly. Just like a bacteria going inside your blood, we call this bacteremia. It's also extremely dangerous to the point that it can kill you. Technically, any infection to the blood, we call this sepsis. So sepsis is very dangerous per se. So local and systemic infections, you have multiple viruses everywhere, that to the point that what's happening with the immune system is spread thin, to the point that you have very little concentration of the immune system going everywhere, and it becomes very hard for your immune system to stop the infection, and this can lead to death. I'm going to give you here a very good example of how a virus really uses a systemic, basically, let's say, infection. So at day 0, you're infected with polio. And usually polio, it happens through ingestion. You basically eat something that has, let's say, polio. It goes inside and it starts to replicate, first of all, in the intestine, because that's the first place it attaches to. It starts to replicate in big numbers to the point now a lot of viruses start to literally go out, because polio, it loves to literally destroy cells. So every time it destroys cells, new viruses are being released. And the first thing is the viruses start to move towards the mesenteric lymph nodes. It reaches the mesenteric lymph nodes and they start to replicate. Again, a lot of death starts to occur and a lot of viruses start to release. And that's where you have the first viremia, viruses going into your blood. And through the blood, they start to really go to all different types of organs. This could be your liver or this could be your spleen. And they start to replicate in your liver or your spleen in big numbers, again, because they're multiplying so much. Polioviruses start to kill a lot of the liver or spleen cells that they release a lot of viruses. The viruses then suddenly go again, because there's so much of it being produced. They enter again the blood. This is your second viremia. And now it basically it passes your blood-brain barrier, this barrier that really separates the blood from our, let's say, periphery, from our brain, because the brain needs to be very clean. because nothing can go inside the brain. It's the most important, one of the most important vital organs that we have. But the poliovirus is able to pass this barrier, infect motor neurons inside your central nervous system. And this is where it can be extremely dangerous, because once it starts to destroy the neurons and specifically your motor neurons, you have paralysis. And polioviruses, as we all know, polioviruses can really kill us through paralysis, to the point that our own lungs stop to work. So polioviruses is an example of a systemic infection. You have other basically infections that are systemic as well, but they are in some sort of way, they do not use the blood. They use something different, which to me is very smart. An example for this is, for example, herpes or also rabies. So let's use this example over here. You have a human that is literally bitten by a dog. The dog, this is a dog that is infected with rabies, is literally going to pass on the rabies to this human. The rabies are literally going to go into the bloodstream, but going to go straight into your, oh, sorry, not to your bloodstream, my bad. It actually goes straight to your muscle. And the muscle, as we know, a lot of muscles, they have these basically neuromuscular junctions, and where you have a muscle here, and then you have a motor neuron. And what's going to happen is literally that your virus, your rabies virus, is going to jump straight into your neurons. Once inside the neurons, specifically this axon terminals over here, we know that a lot of neurons, what they do is they have these massive, basically, let's say, axonal nerves, like these really, really big nerve endings, that they're literally traveling all the way from, let's say, your calf muscle, all the way towards your brain. So what can technically happen is that the rabies virus is literally just going to the track of this neuron. It's traveling through the neuron, and we call this neural spread or neural spread. It doesn't go to your blood, and it doesn't need to. It's just literally going. Literally think about it as a track. It's literally a train track that the virus is using to go to the site that it needs to go. Why would a virus need to do that? Well, it's amazing, because it's now hiding itself from your immune system. It doesn't need it. It basically now this is why rabies are extremely very successful at killing us, because our immune system doesn't even recognize that they're here, because they're inside neurons, and our immune system doesn't come near neurons. It's extremely afraid. It doesn't want to destroy such a vital cell. So the viruses use that to their advantage. Other examples is herpes. Herpes does that. Varicella zoster also does the same thing. So one now, basically, as we said, viruses can do damage quite a lot. But what damage it can do, it could be direct. This could be direct viral damage, meaning that the virus per se itself starts to kill your own immune cell. It starts to kill your own host cell. This could be by, as we said, stopping the mRNA translation. This could be by inducing apoptosis, and etc. You have examples like polioviruses that are literally killing your motor neurons. Ebola viruses are very good at damaging your endothelial cells that are literally in your blood vessels. So your blood vessels have a lot of endothelial cells. You destroy that, well, now your endothelial cells are leaky, and they're literally now spraying blood all over your body. Ebola is extremely known for this kind of systemic hemorrhages. You have literally blood flowing into all parts of your body, into your heart, into your lungs, into basically your liver, everywhere. And it's extremely painful and very deadly. This is why Ebola is very deadly. Other things that can happen is indirectly through immune-mediated damage. So what we see is that we know that our immune system is trying to help us there, but sometimes our immune system could be a bit clumsy in the sense that it can be overactivated. And we see specifically CD8 T cells. CD8 T cells, when we have an immune response against a certain virus, CD8 T cells are usually the main ones that are doing the killing. because CD8 T cells are the ones recognizing MHC1. MHC1 is presenting usually viral antigens. So CD4 is much more differently for, let's say, bacteria. CD8 is much more for viruses. So CD8 T cells, usually they start to kill infected cells, but because they're killing so much of it, they can actually kill bystander normal cells, and this can cause a lot of damage. But again, another thing that can happen is, as we know, is our type 3 hypersensitivity. So this immune complex mediated diseases. So what happens is you have a virus. So this is a virus. So this it says antigen, but this could be a virus that is binding towards a antibody. So you create this immune complex, which is a complex made of literally antibodies and the virus that is attached to. And these complexes, they attach to basement membranes in blood vessels, but they can also bind to literally everywhere, to your liver, to your spleen, to your kidneys, everywhere. And these complexes are extremely immunogenic, meaning that they really will cause your immune cells, specifically neutrophils, to release a lot of cytokines that is really going to cause a lot of death to bystander cells that have nothing to do with it. This happens a lot with hepatitis B. This happens with rubella and parvoviruses as well. One thing that's interesting that we see with viruses is not even despite being the same viruses, let's imagine influenza, not all influenza viruses can do death. Some of them have a increased virulence. And virulence, what we mean by virulence, it's the relative ability for a virus to cause disease. So sometimes we see, for example, let's imagine we have influenza A and influenza B. We see that influenza A, even though they're the same subtype H1N1, this one's H1N1, but they have just some differences, small differences in their genomes, that influenza A is literally killing you, but the influenza B doesn't kill you, does nothing. What happens is that, as we know, viruses really need to have a certain receptors or certain, let's say, proteins that interact with the host proteins, and that determines everything. So sometimes viruses have certain proteins that will literally induce death to a cell. It will literally start killing. Sometimes that virus doesn't have that protein. So virulence is really determined by viral genes. Some viruses have certain genes that can induce cell death. Some viruses don't have that. And what sometimes we can do is that we could use, for example, SARS, influenza, MERS, HPV, Mpox viruses, dengue viruses, all these viruses that if you infect, you can kill someone. But what we can do is we can take these viruses and we can remove these basically genes from them. We can remove a gene that is literally inducing the killing. So these genes that are literally encoding for proteins that are causing cells to die, we remove them. Now these viruses are less virulent. We call these attenuated. So these are what we call attenuated, meaning that they're crippled. They are not able now to cause cell death, and they're literally harmless to us. And we can use them as a vaccine platform now. We can use these to inject. I'm going to be talking a lot about this in my last chapter in vaccinology, of how we can use this for a vaccine. But of course, one thing that viruses need to do a lot as well to be successful, they need to evade your immune system. They need to do that. And the viruses have a lot of ways of evading it. And one of them is virokines. And to me, this is one of the most, let's say, it's a genius kind of way for viruses to kind of hide for our immune system. So what viruses do is they start to produce virokines. Virokines are literally receptors that bind to cytokines like interleukin-1 and TNF. So virokines are receptors that the virus produce. They insert themselves in the host membrane, and they literally bind to interleukin-1 and TNF. But they bind to them. They don't signal nothing. So what they're trying to do is they try to kind of think about it as a molecular sink. They're just trying to sink it, make sure that the concentration goes down so that interleukin-1 or TNF is not actually binding to the true receptor that's causing, let's say, the immune response. So that's how important these virokines can be. Another thing that basically viruses can do is they can snatch down MHC class 1. So as we know, all nucleated cells, have MHC. They need to have MHC1. because all nucleated cells can be infected by a virus. So we always use viral antigens to put on MHC1. This helps our CD8 T cells to recognize them. So here, what the virus is trying to do is trying to make sure that it doesn't get recognized by a CD8 T cell, so it snatches down that MHC class 1. But the virus thinks it's smart by snatching the MHC1. Sure, you might have stopped CD8 T cells, but you've now turned on NK cells. Because NK cells, what they do, usually NK cells, they love to patrol. And if they see a cell that has an MHC, it says like, okay, this is fine. I don't need to kill it because, all nucleated cells have an MHC. But if an NK cell suddenly now doesn't bind to an MHC class 1 on a cell, the NK cell is like, okay, this is weird. All nucleated cells have an MHC. This one doesn't. That means that this cell is either a cancer cell or it's an infected cell. And that kind of triggers the NK cell to start killing. So the virus might be doing a good thing for itself by snatching the MHC1, but it can also be doing damage because now you're activating your NK cells to come in and kill. So NK cells have a huge importance in killing viruses. See the question, 1 second. Because NK cells have a high affinity MHC1. Yes, that's correct, Miriam. To go into more details about this technically. So what happens is that NK cells have, usually they have a lot of receptors. They have inhibitory receptors, but they also have activating receptors. Their name is very self-explanatory. Inhibitory ones inhibit the NK cell to kill, while the activating ones activate. And the MHC really binds to these inhibitory receptors on NK. So it's kind of, the MHC kind of, let's say, protein is literally giving the NK cell the signal that don't kill me through this inhibitory receptor. But when the MHC doesn't have it anymore, you don't have anymore this inhibition, this inhibitory receptor being activated. So now the NK cells that by itself kind of makes it say like, okay, I need to start killing. So yes, they have it. Because of these inhibitory receptors, they have let's say a higher affinity towards them. One thing that we've learned specifically with antigen presentation is that interferons, specifically type 1s, the alpha and beta, they love to literally, when we have a viral infection, interferons are produced to increase MHC presentation, to increase antigen presentation. We need that. So interferons are a very important cytokine that literally puts our whole system on alert for a viral infection. So what viruses can do is they can stop the production, block it, or even what they can do is stop the signaling, the complete signaling of interferon. So now we don't have these cytokines that are alerting us, that are telling our nucleated cells like, hey, start doing antigen presentation. We need viral antigens to be presented on MHC1. Or, hey, we need our immune system to start literally producing CD8 T cells. So that's what viruses can do. They can stop this. And Ebola viruses is very well known to do that. It blocks both the production and the action of interferon. So it's able to stop the production of interferon, but also the signaling. Another thing that viruses love to do is antigenic variation. So either this comes into, let's say, flavors. One is that they have serotypes. So we know that rhinoviruses, for example, have 100 known serotypes. Serotypes literally means they are rhinoviruses, but with literally different but more proteins on their surface. And then, of course, if you're, let's say, infected with, let's say, rhinovirus 1A, sure, your immune system might recognize it, but if you're then now infected with rhinovirus 1B, your immune system is not going to recognize it because their surface receptors or the surface, let's say, proteins are not really, our immune system doesn't recognize them. Antibodies don't bind to them. T cells don't recognize that. So it's a different thing. But other viruses like influenza, HIV, or hepatitis C, they exhibit high antigenic variability, meaning that they start to mutate a lot this constantly throughout the infection. So if our immune system now suddenly has an antibody against this specific, let's say, protein, now suddenly it has a mutation that now it doesn't allow the antibody to bind anymore. Or now the mutation doesn't allow CD8 T cells to recognize it, or CD4 T cells. So this can happen quite either that they have, basically the rhinoviruses has multiple different types already, or that through the infection process, the virus that you get infected with keeps changing. And of course, we have a lot of viruses that are persistent. These are viruses that literally are not cleared by our immune system. They stay there for a lifetime. And for these viruses to really be like that, they really need to have some sort of, let's say, some sort of way to do this. Like, how are they able to stay in our body for a lifetime? Well, either they actually are integrating their DNA inside of us. So now the virus and us are one. Like, as cheesy as that sounds, it's true. So now the virus is literally part of us now because it's inside of our genome. So unless you gene edited it outside, it's going to remain there until a lifetime. Or that the virus is able to evade your immune system extremely well to the point that it doesn't need to actually integrate its DNA inside. It's just there, and your immune system is not able to remove it. Or it replicates in protected sites. This is, for example, herpes. Herpes literally stays inside, let's say, neurons. No one wants to. The immune system doesn't want to kill a neuron. It doesn't want to do that. There's very protective layers that doesn't allow that. So either they use one of these or a combination. For example, herpes does both, actually. Herpes, they integrate their DNA, but they also replicate really well in protected areas. Just for example, other stuff that can actually happen, of course, between the immune evasion is that you can have a rapid antigenic variation. That's a form of immune evasion. But also, some viruses, they spread cell to cell so quickly that our immune system is so hard to catch it. So now let's talk about different types of persistent viral infections. You have chronic state. Chronic state, basically what it means is that the virus it replicates and it sheds for an extended periods. So meaning that the virus infects and it keeps producing viruses for almost a lifetime. The example for this is hepatitis B or hepatitis C. These are classical examples of chronic viral infections. And this really can cause chronic diseases like liver damage or cancer because they're just producing viruses constantly, which really can cause a lot of damage throughout the lifetime. Then we have latent infections. Latent infections are a bit different to chronic in a sense that you have a virus, it comes inside, it starts to literally start to produce a lot of viral copies and do a lot of damage, and then it stops. It goes into this latency kind of step, but no virus is being produced. The difference between latent and chronic is chronic viruses are being produced. Here, no. It stops. Viruses are almost undetected. Suddenly, then something happens. Some environmental stress or stimuli is coming in, and boom, you have viruses start to come up again. So, can be many different things. This environmental stress and stimuli can be many things that can actually happen. We don't know exactly. In the case of herpes, we don't know what these stresses can be sometimes. Herpes does this a lot. It establishes latency in sensory ganglia. There are varicella zoster viruses, and the chickenpox also does that as well. Chickenpox, a lot of us that we have chickenpox, it stays within our lives, and it can be there for a long period of time to the point that you never ever get a single infection or let's say a symptom in your life again. But sometimes in some people, it can sometimes reactivate and we get shingles, and that can be extremely dangerous. And then you have slow viral infections. These are infections that, and generally, they're being produced, but very slowly and gradually, to the point that the first symptom can come up in a year or two. An example is basically what you see in measles or you see with the JC viruses. These, by the way, I'm going to put this in red because I don't want you guys to really know these examples over here. But I know what I need you to know is the viral infections is viral infections, slow viral infections, meaning that they're producing. The virus is produced, but extremely slow. So if you want to put everything on a graph, this is the amount of viruses. This is basically the disease stage or the time, months or years. So acute infections happens when you have a virus coming in and done. There's a recovery phase and that's it. You don't have it anymore. Latent infections, for example, like for herpes, you have a virus infection and then you have nothing. It's undetected. This is why it's this kind of dotted. It's undetected, and suddenly again it pops up. And then this can go undetected and again pop up again. Chronic, you have it, and viruses are still being produced. Still being produced, and basically it can go up or down, depending. Slow infection, the virus keeps literally producing undetectably until it reaches the point that the first symptom starts to pop up because now our immune system is detecting it. So this is how you want to put them all together. Okay, guys. So this has been viral pathogenesis. Is there anything that you guys want me to go over, to explain, or have any interesting questions that you want to ask? Please let me know. Well, if there's no questions, thank you so much, guys, for joining. Good luck on your exam on Thursday. Hopefully, it'll be in person. If not, it'll be online. Depends on if LAU allows basically the campuses to be open again. That depends on all that. Of course, depending on the security measures. I will definitely keep you guys posted as soon as I know what's happening. But if not,
is there a probability that we do it online?
yes, there might be a probability. I don't know still. Like, the thing is that, as you know, how can I put it? The security events are just literally every day they're changing. So yeah, there is a probability. I cannot deny that there might be a probability that we might do it online.
And can we get an email on how the whole format will be if it's online, and how the process will be in the online meeting? I know we need to put two web